Argonne national flaviviruses such as Zika, dengue, and West Nile viruses are arthropod-borne pathogens that present significant public health threats worldwide. In particular, Zika virus recently became an international health emergency, not only because of its rapid spread throughout Central and South America, but also because of its association with severe birth defects and with Guillain-Barré syndrome. NS1 is a highly conserved protein among flaviviruses and plays key roles in viral infections. When the virus infects a host, NS1 is involved in viral replication and packaging, allowing the virus to continue infecting more cells in the host. Some NS1 is also secreted as hexamerase figure 1 into the bloodstream of infected individuals, and higher NS1 levels have been found to correlate with more severe illness. Even in the absence of virus, studies have shown that NS1 alone is sufficient to cause vascular leakage in biological model systems. Researchers from the University of Michigan had previously solved structures for the NS1 protein from dengue virus serotype 2 and from West Nile virus, also using data collected at the GM/CAXSD beamline at the APS. So, when Zika virus infection was linked with an increased incidence of microcephaly and Guillain-Barré syndrome, they realized they could also contribute to understanding this new viral threat. In collaboration with researchers from Purdue University, they conducted studies to determine the structure of Zika virus NS1. Although their previous work with the NS1 protein had established because of its critical roles in viral replication and disease pathogenesis, the flavivirus non-structural protein 1, NS1, is an attractive target for design of vaccines and drugs that could protect against diseases such as Zika, West Nile, and dengue that are caused by flaviviruses. Listed its basic structure, several features were not resolved. Using data collect at GM/CAXSD beamline 23 IDD at the APS, the researchers were able to determine more detailed information about the Zika virus NS1 protein, which helped to fill in these gaps. For example, they found that Zika virus NS1 has electrostatic charge properties that differ from those of NS1 proteins in other flaviviruses. They also identified a part of the protein that was not visible in the earlier NS1 structures, an additional hydrophobic surface for membrane association figure 1 during viral replication. Elucidating the Zika virus NS1 structure helped the researchers to understand how NS1 circulates in the bloodstream of infected patients as a protein lipid particle. According to the researchers, this new information about the Zika virus NS1 structure adds important details of the molecular framework for understanding how NS1 accomplishes its functions in the infection cycle. Science.
The horseshoe crab blood is vital for making vaccines, and now due to the race to combat the coronavirus, these living fossils are hitting the headlines again. The horseshoe crab blood is blue because of its rich copper content, and it is the only known natural source of Limulus amoebocyte lysate, or LAL, which is a substance that detects a contaminant called endotoxin. The crab's blood clots upon encountering endotoxins, which can be lethal if they enter vaccines. Safety tests using the horseshoe crab blood is the industry standard, but environmentalists are advocating the use of a synthetic alternative.
For hundreds of millions of years, horseshoe crabs have been crawling around the ocean floor. Coming ashore to mate in the spring, these living fossils are synced to the rhythms of the tides. Today, migratory shorebirds, sea turtles, and other ocean animals depend on horseshoe crabs for food. They are a keystone species at the heart of many coastal ecosystems from infection. The horseshoe crab has evolved a powerful mechanism for protecting itself from infection. When bacteria sneaks past a horseshoe crab's shell, cells in the crab's blood send out a chemical that clots around the invader, stopping it in its tracks. And it's this property that makes horseshoe crab blood invaluable to the medical
because of
industry.
the absence of observations during the transient activity time. Supported by the above two cases, we suggest that they are correlated to sources of cometary
to make sure that
activity.
they're not contaminated.
4.4.
catch and release
Clusters of
system
patches. There have been
supposedly leads to
several
few crab
observations
deaths.
of
So while
clusters of bright
Polymyx and
patches that are
antibiotics are one
individually
of our
smaller
last
in
lines of
size
defense
compared
against
to the aforementioned
multi-drug-resistant
features.
bacteria. They are made
These
by
clusters have
certain
mostly been
gram-positive
observed in the
BAC
pre-perihelion
to selectively kill
period and they are
gram-negative
located
bacteria
near
by
cliff
disrupting their
structures.
outer membranes through
Therefore, these
interactions with
clusters could
specific
be related to cliff
lipopolysaccharides,
collapse events, in
LPS.
which the
However,
resulting
in an
debris
escalation of
exposes
this
water
conflict
ice
and in
contained and
response to
preserved
our use
inside
of
since their
polymyxins as
formation, as the
antibiotics,
major
some
cliff collapse in
gram-negative
Aswan
bacteria
area
have
has
developed
been
resistance
explained
to
by Pajola et
polymyxins.
al.
This is predominantly accomplished by an
2015-2016. Figure
amino-arabinose
6 shows a
transferase
cluster of
ARNT
smaller
that can
bright
modify
features
the
in the
LPS
Kepri
so that
region.
polymyxins cannot use
First
it to bind
observed
to
at
the
the end of
membrane
August
anymore. Drugs that
2014,
inhibit
this
ARNT
cluster
could
remained
overcome
very
this
stable
resistance
for
and
about
improve
3
the
months.
efficacy of this critical
We
class
numbered
of
some of the
antibiotics.
individual
In an
patches to
important first
emphasize
step
their
toward
stability
developing
in the
such
sequence of
inhibitors, a
images.
research team utilized
This
high
cluster
brightness
might have
X-ray
remained
beams from
stable
the
for
APS
longer,
to
but it
solve
is
the
difficult
structure
to
of ARNT
conclude
with
owing
and
to the
without one of its
inadequate
lipid
temporal coverage of
substrates. The work
images.
reveals how
An
substrates bind to
observation
the
dating
enzyme
back
and
to the
identifies
25th of
amino
March
acids that are
2015
critical for its
indicates that the
catalytic
entire cluster
activity.
has
Important
sublimated away,
information
which
for drug
indicates the
design.
influence of
Argonne National
increasing solar
Laboratory
irradiation on the clustered
153.
individual
Polymyxins
patches.
bind to LPS on
This
the
cluster was
bacterial outer
firstly
membrane
studied by
through
Pomerol et
electrostatic
al.
interactions with
2015
negatively
and
charged groups
Baruchi
on
et
the
al.
LPS.
2016
ARNT
attributed its
modifies
presence to
LPS and
H2O
confers
using
resistance to polymyxins
VIRTISM
by
infrared
catalyzing a
spectra.
reaction that caps
Frost
LPS
frost
with
has been
supposed
observed
negatively
since the
charged
arrival
sugar
of Rosetta
L-araflor,
de
and the
Sanctis
sugar
et
is
al.
added to a part of LPS called
2015 at
lipid specifically
67
at the negatively charged
3.6
groups that
astronomical
interact with
units by
polymyxins.
VIRTIS
L-araflor and is
spectro imager
transported
and
to
has
the
since been
reaction by
continuously observed by
undecaffeinylphosphate
OSIRIS up to the end of the
lipid
mission.
carrier. This means
3.8
that ARNT must
astronomical
bring two
units with a
lipid
pronounced
substrates,
abundance
lipid A
towards
and
the
the
perihelion
L-araflor,
passage
and
for
together
Nazier
to
et
the
al.
active site where catalysis to occur. The
2016.
team,
This
led
frost
by
could be
Columbia
observed at the
University
local dawn on the
society
comet,
scientists
where the
working
sunlight
together with
returns
colleagues
after
from
about
the
6
University
H.
of Georgia,
Thus,
the Medical
the shadows
College of
cast by
Wisconsin,
different
Cornell
morphological
University,
structures
the New
get
York
shorter as the
Structural
sun moves
Biology
towards the
Center,
local
and the
azimuth,
Technische
revealing the
Universität
frost
München,
formed over the
Germany,
cometary
solved the
night due to
crystal structures
condensation
of
of
ARNT and
previously
ARNT plus
sublimated
UNDP
water
at the
ice.
NECAT.
The frost could be observed for some minutes before being rapidly sublimated away, depending on the heliocentric distance of the comet. This process continues as long as the solar irradiation is strong enough to sublimate the frost. Once the comet is beyond the current snowline of the solar system, the diurnal water cycle of the comet ceases, leading to no further frost sublimation until the comet returns inside the snowline for its next perihelion passage. This diurnal cycle of water was more pronounced as 67 P got closer to the perihelion, as water production rates on the comet rose due to the increasing solar irradiation. Hence, close to the perihelion, both inbound and outbound, frost was ephemerally visible on a given location during the local cometary morning. It is also possible that the 4 types of bright features introduced earlier contributed to the diurnal cycle of water on the comet as they have been constantly sublimating, thus liberating the amount of water ice necessary to sustain the diurnal cycle of water on the comet.
Specific genes that indicates the presence of life to the organism. 14 IDB by AKRS Life Sciences. Phytochromes are composed of evolutionarily conserved domains. The per/ARN/SIM PA and phosphodiesterase/adenyl cyclase/FHLAGAF domains assume a tightly packed globular structure stabilized by a figure 8-like knot formed by Argonne National Laboratory 166 APS Science 2006 Argonne National Laboratory 165. Most enzymes have one job. 21 years on from those first X-rays, we like to think we have this light source business figured out. Most enzymes have one job, with structures that are honed by evolution for a single specific purpose. However, a few special enzymes do double duty, targeting 2 different substrates. Such enigmatic catalysts beg the question of how one enzyme with a single structure recognizes 2 substrates with different structures.
Their impacts on government, writing, and cultural trends are part of our everyday society. But what about the Phoenicians? You might not have heard of them, but their many contributions are just as essential for us as other bygone civilizations. From around 1550 BCE to 300 BCE, the Phoenicia Empire had a significant civilization built primarily on shipping trade throughout the Mediterranean. They established coastline cities that allowed their people to have a rich, enterprising maritime trade culture. They created waterways of business for many of the civilizations at the time, making it possible for someone from Greece or Rome to communicate and trade with people as far away as Britain. The Phoenicians didn't have a centralized location for their government. Perhaps the city-state of Tyre came closest to their established authority. As an alternative, like many ancient ruling powers, Phoenicians established independent city-states, a single city with surrounding dependent territories along the Mediterranean Sea. They used strategic city settings that allowed them a stronghold for maritime trade routes. Lebanon, Syria, and most of northern Israel, as we know them today, grew out of the Phoenician city-states, and the ancient city-state of Tyre is considered the most prominent Phoenician establishment with over 30,000 citizens at its height of power. Phoenicians weren't interested in metal crafts, writing in scrolls, or art, unless it had something to do with shipbuilding. Their entire society grew around their understanding of the Mediterranean Sea. That means that if you wanted to set up a trade route over water in 1550 BCE, you had to use Phoenician ships.
When people think of Israel, volcanoes are not the first thing that comes to mind. Medina in 1250 and the volcanoes which recently erupted, such as Saudi Arabia. One of its volcanoes, Harat Rahat, nearly destroyed the city of Medina in 1256 when it covered a vast area with lava. Another volcano in Syria, known as Safa, erupted quite recently in 1850. With all of this being said, Israel also contains an active volcanic field. The volcanic field in question is known as the Golan Heights, which is located in the northeast section
on papyrus and have
from memory, enter at the DOS prom /gold key d Kenneth deckpad.key and g0ldkey.com version 2.0
is comprised of 3 large sectors. The main sector of the settlement, the Acropolis, located in the
was king. He ruled 36 years. You so it's roughly 27 years ago.
As he did not review King Uzziah, Uther, and the first
stones placed here by an ancient tribe who came to
F/CPRISI $7F The affinity of immobilized enzyme towards starch was higher compared with soluble enzyme. We have estimated 0.1.146002 3.95. The experiment shows that aerobic denitrification in this soil-derived community is insignificant without prior exposure to an anoxic phase. The role of NO2 as an inducer of aerobic denitrification is unclear. Cells which are exposed to short anoxic periods, for instance created by flooding and subsequent drainage, will probably keep on denitrifying even once the O2 supply is partly or fully restored.
Seth and Ash include contributions in the line of sight of the
D20 content ppms, D20 deuterium content of waters in various parts of the world, D20 content ppm sample, Oceans North Sea 161.3, Atlantic near Quiberon 162, 8 Atlantic near Long Island 159.9, Antarctic number 1 157.1, Antarctic number 2 159, 8 Antarctic number 3 160.9, Indian Ocean 161, 0 Lakes and inland seas, Dead Sea, Baltic Sea, Mono Lake, USA, Kanujärvi, Finland, Lake Geneva, Loch Ness, Lake Michigan, Rivers Weser, Rhine, Po, Niger, Ijoki, Finland, Vienne, France, Iskre, Springs Il Arian, Tunisia, Mineral Springs of Tibériade, Caldanaxia, France, El Imans Tir, Alps, 162.6, 156.1, 152, 8 146, 8 147.5, 154.5, 153, 2 152, 0 151, 7 149.4, 157.7, 147, 0 153.3, 3 to 4 p.m. respectively. Overall, the ground level ozone concentration displayed a pronounced spatial gradient, increasing from the south to the north of the region due to the differences in vehicle exhaust emitted VOx and PM2.5 concentration between northern and southern Beijing. A major value of NOHO2 N2 that is consistent with Rosina measurements of NOHO N22 Beeler ETAL 2015.
Damage as well.
It's trapped inside of the ice. We found a living sin. We can bring it back to life. If we can find the mechanism how that bacteria stays alive, then we probably we'd be able to find a tool to prolong our own life.
This from Moscow. If you leave the main highway connecting Moscow and St. Petersburg for a while, what would you see? Thriving agriculture? Cottage communities? Thick forests? No. There's a good chance that the first thing you'll see will be the tall, mighty plants, like a figment of some science fiction writer's imagination, that grow along every single row. But it's nothing too scary, right? Some kind of native flora. Well, yes, it is now. Only problem is that flora isn't supposed to grow there. It shouldn't grow. It grew by 3 to 5 centimeters per day. It is now. Only problem is it was cut off in time and used as animal feed, and take root in the soil very well. And since scientists were constantly breeding new strains, they were also able to improve these qualities. For example, the number of seeds increased tenfold. Low northern temperatures or the lack of a long daylight and bright sun did not hinder the hogweed.